Assalamu alaikum everyone. My name is Dr. Evelyn Saba and I am serving as Assistant Professor for Veterinary Physiology in the Department of Veterinary Biomedical Sciences, Faculty of Veterinary and Animal Sciences, Pir Meher Ali Shah Arid Agriculture, University Rawalpindi. It's a privilege for me to present the research from my lab in this prestigious conference, that is International Brain and Biomedicine Conference. Today, the topic which I will be discussing with all of you and sharing my research, that is the anti-inflammatory activities of cinnamon, henna, and turmeric in chicken peritoneal macrophage. Even though that this topic really does not relate with that of the brain. However, because the theme included veterinary sciences and medicine, so therefore I am here to present my work on this topic. So the main focus of the research is actually inflammation. Inflammation, most of you may know that it is a biological response which is triggered by many factors. In fact, it is actually the natural response of body towards a foreign invader, and it is characterized by 5 cardinal signs which we mostly say, that is redness, loss of function, pain, swelling, and heat. So there can be 2 types of inflammation, one which is short lived, that is acute, and the other one which may have a long time, that is chronic inflammation. So for the treatment of inflammation, normally there are 2 set of drugs which are mostly used, that is steroidal anti-inflammatory drugs that are called as SEDs, and non-steroidal anti-inflammatory drugs that are called as NSAIDs. And here in the table you can see that there are different set of drugs which are actually available over here for the treatment of inflammation. Now, given that there are different set of treatments which are actually available for the treatment of inflammation, that is the SEDs and NSAIDs, even though that they have lots of effects which are good, that is why they are being used, that is anti-inflammatory and analgesic and anti-arthritic and so on. However, dependence on these drugs, which happens most of the time, it may lead to a lot of side effects like hepatic and GIT dysfunctions, cardiovascular, and even cerebral complications. So, what is actually solution for that? How we can actually use something which will be anti-inflammatory as well as it may not also give lots of side effects? Because having said that, inflammation is actually going on in a very quick and almost every time in the body. So dependence on SEDs and NSAIDs is very natural, but there can be a shift towards the alternative therapy to reduce the use of these SEDs and NSAIDs. And having said that, there has been a lot of shift towards the alternative or the herbal therapy since past lots of years. Why? Because they are mostly non-toxic, even though that there are some compounds in these things which are toxic. However, broadly, they are almost non-toxic. Plus, they are easily available, and also they can be sourced locally. In fact, I would like to say here that most of the allopathic drugs or the synthetic analogs that we used, actually they are the copies, or you can say they are the chemical alternatives of those single compounds which are present in these drugs. So in my research, I selected 3 commonly available herbs and spices, that is turmeric, henna, and cinnamon, because all these 3 are very much locally used in our culture and in our country. So it is very easy to procure these compounds and they can be likewise used. So turmeric, as we all know, Urdu में इसको हल्दी कहते हैं, and the scientific name is curcuma longa. And everyone already knows that it is having a lots of anti-inflammatory effects. The second one was cinnamon. इसको उर्दू में दालचीनी भी कहा जाता है, and this is also a very commonly used spice, and also it has reported potent anti-inflammatory effects. When we come to henna, henna को उर्दू में मेहंदी भी कहा जाता है, and this is mostly applied for dyeing purposes and also for cosmetic purposes. However, in the pure form, henna has got lots of anti-inflammatory effects. Now, why I used these ones, as I told before, that they are very much locally available, and I actually wanted to check their effects in the inflammatory model, and for that reason I actually used chicken peritoneal macrophages. So material and methods included all of these. As you can see over here, sample was collected and then their extracts were prepared, birds were procured, and sheep red blood cells were prepared, hyperimmune antiserum was prepared, then the macrophages were isolated, and then they were um, given the sheep red blood cells, and we checked their phagocytic activity. Then we also checked either these extracts, did they show any kind of cytotoxicity, and for the anti-inflammatory effects we checked the NO assay, and we also checked the expression of genes which are related with the inflammatory pathway. Finally, for the significance, we used statistical analysis. So what the results of this study showed, as you can see here, that henna, cinnamon, and turmeric, all three of them, actually we used two doses of both of all three of these herbs and spices. So the highest doses, they phagocytosed most of the sheep red blood cells. It showed that these extracts actually, they enhanced the phagocytic activity of the macrophage. I mean, normally macrophage do phagocytose, that is a part and parcel of their function. However, when we treated these cells with the different spice extracts, their phagocytic activity was increased. The arrow in these pictures is actually showing the RBC, and the other remaining cells, you can say they are the macrophages. And for the cytotoxicity, we did not find any cytotoxicity for these extracts, as it is shown by this MTT assay for various doses. And for the nitric oxide production, because it is nitric oxide is naturally produced as a result of inflammation. However, its overproduction may lead to the signaling of the inflammatory pathway. So for any extract or anything that has to show its anti-inflammatory activity, it should decrease the NO. So what we find here is that the extracts at different doses, they decrease the NO activity. And further we move towards the expression of inflammatory mediators and cytokines. So INOS and COX-2 are the mediators, whereas TNF alpha, interleukin 1 beta and 6 are cytokines. So as you can see that all mediators and cytokines also were decreased with the treatment of these extracts. And finally, we found out the pathways which are actually involved for the inflammation. So there are two pathways, that is NF-kappa B and MAPK. So they are the classical pathways of inflammation as well. So we can see in these graphs that these extracts not only decrease the expression of NF-kappa B, but also the different subfactors of the MAPK, that is ERK, JNK, and P38. So overall, we can say that the inflammation was induced by the classical NF-kappa B and MAPK pathways, and it was reduced by these extracts. So as you can see here that there was a good anti-inflammatory activity, but in our study we found that turmeric was actually very much potent. Among all three of them were good. However, turmeric was most potent. So what we suggest is that in order to bring these spices for animal use, there has to be in vivo trials, and Further, later on, there can be formulation of different drugs or different kind of uh, treatments for use in the veterinary. So that is it from my side. And these herbs and spices overall, they're very good for inflammation overall. And not only for, you can say for peritoneal, but for the overall inflammatory conditions in the body. So they can be very much inculcated in the normal diets of animal as well as the human beings. Thank you very much.